Good afternoon, everybody. We're going to be talking about Chapter 4, which is looking specifically at the structures of prokaryotic organisms, bacterial and archaeal cells. We're going to show the different groupings of these organisms first. Then we'll talk about how the structure of the cells works. So I've already mentioned that we group all living things into categories depending on the characteristics. And typically, now that we have the means, we use chemical characteristics like DNA and RNA to help us to group these organisms properly. There are multiple, that sounds silly. So I'm going to say there are millions of bacteria and archaea that fall under the domain bacteria and archaea. And within bacteria, we have a lot of different phyla of organisms. In archaea, we have basically 2 phyla of organisms. So the ones that we care about most are the proteobacteria, the firmicutes, actinobacteria, cyanobacteria, chlamydia, and spirochetes for our organisms that fall under the domain bacteria. Proteobacteria are the largest group of bacteria, and they include organisms that are found in our gut. So E. coli, a lot of organisms that can be found in our gut are enterics. So E. coli is an example of an enteric bacterial species. Serratia is another one. Then we have the firmicutes. Firmicutes are the majority of, or I shouldn't say the majority, but in our lab, the majority of the organisms that we deal with that are gram-positive are firmicutes. So we will work with Staphylococcus, we will work with Enterococcus, which is very closely related to Streptococcus. We play with Bacillus. These are all firmicutes. Actinobacteria are important because they help in producing certain antibiotics that we use to treat different infections. Cyanobacteria are really important because they are photosynthetic bacteria that are very similar in activity as our own algae, but they are bacterial species. They are not eukaryotes. Chlamydia are important because they contain very pathogenic tiny bacteria that have to live inside of your cells. So a lot of people look at chlamydia almost as if it were a viral or structure because they can't function outside of the cell. But that doesn't mean that they are viruses. They are obligate intracellular parasites, but they also are living organisms that have their own DNA. They can replicate on their own, or they can replicate inside of our cells. And they have peptidoglycan, they have the cell structure and everything. And then we have the spirochetes. Spirochetes are beautiful bacteria, but they can cause some really dangerous diseases. So one of the best examples is Treponema pallidum, which produces the disease syphilis. Under the domain Archaea, we have 2 large groups, Euarchaeota and then Crane Archaeota. Euarchaeota contain our methanogens, which are organisms that cannot live in an oxygenic environment, so they live in an anoxygenic or an anaerobic environment. We have halophiles. Extreme halophiles are organisms that can live in very salty conditions, above like 10% salt conditions. So think about the the Dead Sea, bacteria that are growing in the Dead Sea. That would be an example of a hyperthermal or an extreme halophile. Hyperthermophiles are organisms that live at very high temperatures and very low pHs. So, organisms that live in at the I cannot think of it. Hydrothermal vents. That's what I wanted to say. Hydrothermal vents. That would be an example of a hyperthermophile. The Archaea, these are organisms that grow in very acidic, hot environments. So like volcanoes would be an example. Bacteria and archaea, so prokaryotes, one way we can help to separate the organisms is by their structure. So all organisms have their specific structures that helps us to identify them. If we see a cat and a dog, they both have 4 legs. They have very similar DNA, but you know the difference between a cat and a dog because they look different. Same with bacteria. The most common shapes for bacteria are bacillus-shaped or rod-shaped and spherical-shaped or coccus-shaped. Please note that when you say the word bacillus-shaped, that's not talking about the organism. Some organisms are named for their shape. So Bacillus cereus, which we have in our lab, is named for its bacillus shape. Another type of structure that you can find are spiral-shaped organisms. We have one in our lab called Vibrio. Vibrio is a spiral-shaped organism. It's actually, I don't want to say it that, oh, sorry. Vibrio is a curved rod, so not so much spiral-shaped, I'm sorry. Spiral-shaped, it's under this group of spiral-shaped, but it's more of a curved rod organism. So you're not going to see these beautiful like spirochetes or these big, long spiral, structures. It's more like that curved, that comma-shaped organism, okay? If we have a bacillus-shaped organism, they can be attached together. So they're growing, and when they replicate, they stick together. And this forms a streptobacillus. There's a diplobacillus. We can also see that in our coccus-shaped organisms. Here's a diplococcus, here's a streptococcus. Organisms that are coccus-shaped can grow in clusters. This forms what we call a staphylococcus, or a cluster of coccus cells. And one of our species produces this tetrad. Tetrad is 4 coccus cells together. This is very common in our micrococcus species. Prokaryotic organisms are organized very much like eukaryotic organisms. We have internal structures that are put into compartments, or they're put into one region and nothing else goes there. So they are organized both externally and internally. Inside, we have a region where we have our DNA. So DNA is found in a region called the nucleoid, which is found in the cytoplasm. There's nothing surrounding the nucleoid, but that's where all the DNA sits. We have ribosomes floating all over in the cytoplasm. We have these tiny structures. These are extranuclear DNA called plasmids. We have tiny little sacs or storage compartments called inclusions that can store nutrients, waste products, gases, depending on what we might need. Then we have the cell wall and the plasma membrane first, and then the cell wall. plasma membrane of bacteria is similar to the plasma membrane of the eukaryotes. So when you took general biology and you learned about the plasma membrane, there are a lot of similarities. They have the same function. They're made up of mostly the same structures, but there are some differences that make or that help us to differentiate between eukaryotes and prokaryotes. The cell wall. Almost all bacteria have a cell wall. The cell wall contains proteins and carbohydrates, and that's one of the major things that helps us to differentiate bacteria from archaea from eukaryotes. And surrounding the cell wall, a lot of bacteria have a sugar coating or a glycocalyx. Glycocalyx can be found in the form of a capsule or a slime mold. Slime mold, is that what I want to call it? That's what I'm going to call it for now. Okay. The capsule is very organized. In both cases, though, this protects the cell even further. And then growing out from the cell itself, from the cell wall, you can have flagella that help for movement. You can have pili that help to allow the cell to stick to surfaces or to attach cells to other cells. So pili are important to bacteria, and they're only found in bacteria. They function to allow cells to stick to surfaces. Pili help us to form biofilms, because once we have a sterile or an environment that has nothing on it, the cell can use its fimbriae or pili to attach. We have special pili called conjugation or sex pili that allow us to attach one cell to another cell to allow exchange of genetic information. Flagella function and movement of the cell. Eukaryotic organisms can also have flagella, but the movement is going to be different for prokaryotes and eukaryotes. For prokaryotic organisms, you have a series of runs where you're moving in a specific direction and tumbles where you're kind of bouncing around. In eukaryotes, we typically just have movement towards or away some attractant or something that is going to potentially cause harm. So here you can see movement, a run here, and then a tumble. And then you have a run and then a tumble, and a run, and then you'll have another tumble. The more attractants we have, the longer the run will be, and the less tumbling we will have. If you're interested, there's a video here that will go through the movement of bacteria using their flagella. The glycocalyx is a sugar coat that surrounds the bacterial cell. It can be found as either a capsule or a slime layer. In both cases, its function is for protection, and it also helps to adhere the cell to other environments, helping us to produce, again, that nice microbe biome, where we have lots of microorganisms living together in like a plaque. So the cell wall of the bacteria is a protective structure that almost all bacteria have. In general, just assume all bacteria have a cell wall. There's like one that doesn't, that I know of. And its function is for protection, but it also helps to maintain water balance of the cell and cell shape. So the cell wall of gram-positive and gram-negative organisms differs. You're going to learn how to do gram staining this week. And one of the things you'll notice is that gram-positive bacteria can take in more stain than gram-negative. The reason is because they have a differing amount of peptidoglycan. Peptidoglycan, which I tend to write down as PTG, is only found in bacteria, so you'll never see it in Archaea, and it's found in a thicker amount in gram-positive bacteria. There's less peptidoglycan in gram-negative bacteria. There's still peptidoglycan, but just less of it. So here we're looking at gram-positive structure. Here's the plasma membrane, and then here's the cell wall, and there we have this thick layer of peptidoglycan, and within the peptidoglycan we have all of these teichoic acid molecules, which helps to give gram-negative bacteria a negative, or gram-positive bacteria a negative charge, along with the proteins, and that helps to pull in more of that basic stain. Gram-negative bacteria have a thin layer of peptidoglycan, so you can see that right here. But gram-negative bacteria are more complex. Since they don't have a lot of peptidoglycan in their cell wall, they have instead a second membrane. So they have their normal inner membrane right here, this plasma membrane, and then they have this outer membrane as well as this peptidoglycan layer. The outer membrane contains an O polysaccharide and a lipid known as lipid A. This is called a lipopolysaccharide altogether. So this lipopolysaccharide is very important. Lipid A is a molecule that our immune system recognizes. And so it actually has influence over our health. The lipopolysaccharide, lipid A, and the O antigen is known as an endotoxin and can actually cause a lot of problems in our cells. So when we have this, or what the endotoxin does, when gram-negative bacteria are killed off, the endotoxin is released into our blood, and it moves to our hypothalamus. And for those of you who haven't had anatomy and physiology, or maybe have forgotten this part, the hypothalamus is one of our very important homeostatic regions of the brain. And it helps us to maintain a lot of different processes, including body temperature. And lipid A, or the lipopolysaccharide, moves in, and it basically tells the hypothalamus to reset its temperature at a higher temperature, causing our body to heat up. So this gives us a temperature. The more gram-negative bacteria we release, the more of the lipopolysaccharide that is released into our blood, and the more times our hypothalamus is told to set our temperature higher. This can cause us to get a very high temperature, which can lead us to endotoxic shock, severe problems. The O antigen alone is actually useful in helping us to identify strains of different species. So think about the E. coli. E. coli that live in our gut are beneficial E. coli, but there are pathogenic strains of E. coli that can get into our system as well. So let's look at the cell wall and how different drugs might affect it. Actually, we really look at two drugs, penicillin and lysozymes. Lysozymes are made by our bodies. Penicillin is made from the mold Penicillium. So what penicillin does is it binds to proteins in the cell, and it inhibits peptidoglycan synthesis. And so in gram-positive bacteria that have a very thick layer of peptidoglycan, lots of layers of peptidoglycan basically, penicillin can be very detrimental because without the cell wall, we don't have structure and we don't have protection from osmotic conditions. And so when it binds to the cell wall proteins that produce peptidoglycan, we can't produce peptidoglycan, and the cells are going to die. So it's going to be very effective against gram-positive bacteria because positive bacteria have a thicker layer of peptidoglycan. Gram-negative bacteria have thinner layers of peptidoglycan, so it doesn't do as much because they have that second cell membrane. And this is just a fun little cartoon, the bold and the bacterial. I don't care if he is penicillin. We're in love. The bacterial cell talking, and you know, penicillin and bacteria, they're going to die. It's kind of Romeo and Juliet-ish. So hopefully you found the humor in it. I always do. I'm smiling over here. So I'm going to stop here, and then I'll pick up on this in the next video, okay? Bye-bye.